All right, good evening, everybody. Thank you so much for coming out tonight, and those who've zoomed into the meeting for us. Whoever hasn't grabbed food and everything like that, just be respectful in coming back in. I appreciate that. And then anyone in the Zoom meeting, if you could please make sure that your video is closed and that your audio mic is silenced for the majority of the lecture, that would be fantastic. So it's my honor to introduce our guest lecturer coming from all the way from Boulder today. Dr. Kristi Anseth is the Distinguished Professor in Chemical and Bioengineering and is the Associate Director of the BioFrontiers Institute, and she's here to speak on all the wonderful and cool work she's doing up there. Okay, thank you.
Well, thanks for the invitation to come and speak with you all today. I understand that it's a fairly diverse audience and activities that go on in this department. And so what I tried to do is hopefully talk about some different projects that we've been working on integrated over time and some of the ways that we think about it. And I tried to pick some that have some more of a translational bent as well. And so let me begin by just saying I'm a bioengineer. And so I work in this space where we try to develop technologies that can be useful to hopefully address medically important problems. And one of the areas that I've been very interested in is an area that we might call tissue engineering. Others might call it regenerative medicine or cell-based therapies. So I thought I'd just begin by give some background. And one introductory slide, this is actually quite old now, but this came out in JAMA, in the Journal of American Medical Association, right when 1999 was turning to 2000. And it's kind of an interesting little editorial, but it's talking about what all the children born during this turn of the millennium, what their lives would be like. And so in one part of this article, it talks about, right, so JB is now a 150-year-old female and talks about her past history and all the diseases she's had. She's still alive, and coronary bypass grafting 80 years ago, gene therapy. Here's one I like, that bilateral knee replacements, but also femoral head excision with regrowth by a growth factor for treating osteoarthritis, right? So it's talking about all the technologies, and it's a little bit just more entertaining. It was fun to read. But so in my world, I'm interested in, and I think a lot about working with different types of cells and how we can use them as therapies to get tissues to heal themselves when processes go awry. And as you probably all know, the topic, our tendency of using cells to improve patients' quality of life and to treat them has been going on for quite some time. Right? So blood transfusions and bone marrow transplants save many people's lives. The Edmonton Protocol, where they transplant islets from a donor patient to somebody that has diabetes that's very difficult to control and regulate. One that I'll tell you a little bit about is autologous chondrocyte implantation. That was one of the first cell-based therapies where they took cells from patient-specific cells. And they had to go through all the approval of how could I expand these in a way and then deliver them back to patients with the hopes of trying to regenerate their cartilage instead of having replacements or implants. And in fact, if you go to the clinical trials website and you just search on all the different clinical trials that use stem cells or cells, there are tens of thousands of hits. And these go from anything of injecting cells back into patients' hearts after they've had a heart attack to delivering dopaminergic neurons to patients' brains that have Parkinson's disease, or immunotherapies, and CAR T-cell therapies is really prevalent right now. But one of the ways that we think about this is as this continues to grow, a lot of these treatments are suboptimal. Some are more successful than others. But in many cases, when they inject these cells in these clinical trials, maybe 1% to 2% of the cells engraft and survive, a very small amount. And so from our perspective, that makes us think about, in our bodies, cells reside in tissue niches. And so from a bioengineer's perspective, we think about a missing part of this might be, can I design synthetic ECM niches, biomaterials, when I'm delivering these cells so that perhaps I could improve survival, improve outcomes. And so what I wanted to tell you about a little bit in the beginning are some of the biomaterials that we work on, and then I'll get to the applications. This is the first time I gave a seminar. I had to give learning objectives, so I wasn't quite sure the format. So I'm going to teach you a little bit about some of the biomaterials that we work with. So the idea and the way that we think about it is we think in all this space outside of a cell. So we think of the extracellular space when we're designing different types of biomaterials, and we try to think about what information do cells get from that extracellular space. And if I could understand that, could I design better types of biomaterials? And better types of biomaterials might be coaxing those chondrocytes to regenerate and repair cartilage. It might be giving a survival cue for a stem cell that I'm trying to deliver into an inflammatory region. And so rather than those cells dying, can I protect it? Can I give survival cues? And the part that I'm not going to talk maybe about as much today, but part of this, sometimes we don't know what information is needed. So we also spend a lot of time developing better types of biomaterials for in vitro culture. So it's not just on a plastic dish, but I can watch cells in something that hopefully better recapitulates what it would be in vivo, and then I could maybe learn something about controlling signals and which signals I should present to cells and how cells receive those signals. So there are all kinds of different types of biomaterials that you can pick. And one that my group does a lot of work on are these hydrogel materials. So that's a macroscopic picture of a hydrogel. If you could see the molecular level details of this, what this would be is it'd be long macromolecules, long chains, that normally would just dissolve in water. But because of interactions between those chains, they form this mesh or network, and it renders them insoluble. So what happens is they imbibe large amounts of water. It's a solid, a gel. And so what's nice about this class of materials is that it's very similar to many of the soft tissues in our body. So this high water content gives them mechanical properties that are similar to much of their native environment. The high water content allows transmission of like cell-secreted cues if they want to signal to one another, or if it's going to regenerate and regrow a tissue, it allows very facile transport, is what we call as engineers. So then sometimes the key part is which signals should we be delivering, and that spans from the molecular scale of how is the cell going to bind to this, how might it move through this at a mesoscopic scale, all the way to a macroscopic scale if I want to make a tissue of a defined shape. All right, so they're also really nice for imaging, I've mentioned. So these are fluorescently labeled chondrocytes that are embedded inside of this gel, and so it's very easy to put on different types of microscopes and renders these culture environments very amenable to three-dimensional imaging. All right, so here's just a tiny bit of teaching. So when you make these hydrogels then, so when we think about designing them for an application, we think about, what should I make them out of? Should I use synthetic materials? Or should I use natural materials like collagen or hyaluronan? Or should I use a synthetic material like a polyethylene glycol, which I'll tell you about? And there's pros and cons to each of these. Natural materials are nice because many of them are found in our body. But typically when you buy these or use them, they don't have the same properties as the collagen in your body. But cells get lots of signals for them. The synthetics are nice because I can make them and I can make them reproducibly. And when I make them one day, I can make them the same, months from now. And I can also, I have a bigger array of chemistries I can choose from. And then I told you that you have to cross-link them, and so then there's different ways. So sometimes we make covalent links, we chemically cross-link them. That makes them stronger, so if you have a load-bearing application. But other times they're just physically associating. There's interactions between them when you put them in water, like hydrophobic regions that'll interact together. And what's interesting about those is that helps make them injectable because I can disrupt those bonds under a load. So there are different types of ways that we pick this. And then how you shape it or form it. Do you want it to be a solid like this? Do you want it to be a bunch of particles that will assemble together and make something porous so cells might infiltrate? And then in many cases, we want it to break down. So we don't want it to be permanently left in the body. So how it breaks down, is it hydrolysis? Water will just cleave it over time with predetermined kinetics? Or is it going to be some enzyme that's in the body and that the cells will make and degrade? So those are some of the things we think about. And so here's probably some common hydrogels you see. So contact lens is a covalently cross-linked hydrogel. Super absorbents. So sometimes these are also used like emergency clotting types of materials, but they're also used in baby diapers and things. They swell up thousands amounts of their weight in water. It's quite remarkable. So they're also really good for drug delivery systems too. Embed a drug, and then when it swells, it releases. A lot of them are used in wound healing to control the wound environment. And then, as I mentioned, they're kind of used in this area of cell delivery vehicles and growing tissues in these types. So these are just some of the applications of hydrogels. I also threw together some statistics. You may not be able to see this very well, but if you look at all the applications of hydrogels approved for products, almost in a volume way, almost all of them are related to the eye, not just contact lenses, but things that you put inside of your eye, endocapsular lens replacements. But there's also some used drug delivery for cancer, a lot's used in dermal space for wound healing, and also as fillers and different types of plastic surgery types of treatments. If you take out the ocular, because that dominates so much, you can get an appreciation for all the different areas that hydrogels are currently used. And then, depending upon the hydrogel you pick, they're put together in different ways, where it's either just like I don't know, if you've made a hydrogel, so if you've made Jell-O before, that's a hydrogel. So it might just be a bulk hydrogel like that you'd shape in a certain form. A lot of times they'll have drugs inside of them, so they're drug-eluting. So they're good for controlled and localized delivery. Other times they're like a patch, a thin film. Some of them even have shape memory to them, so that they can change shapes. All right, and so then the last thing is that there's also quite a few of clinically approved injectable hydrogels as well, and several used in clinical trials. So some of it depends upon the application, the way you need to process it and deliver it, and do you want to put drugs in, do you want to put cells in? Okay. So I think it's about the last background slide. So I think I mentioned some of these already, but some of you may have experience, but with the natural gels, probably collagen, fibrin glue, it's a product that's formed that mimics the blood clot. Hyaluronic acid, hyaluronin, Matrigel is used a lot, especially in the stem cell field. They grow miniature organoids in Matrigel quite a bit. That's just secreted by a cancer cell line. And then they take the matrix of what the cells secreted. There are all different types of cross-linked gels, and there are synthetic ones, that, like these PEGs and polyacrylamides. Alginates come from seaweed. So in those, again, I don't expect you to necessarily read all the details of this, but I just wanted to give you an appreciation that there are cancer products that use PEGs, and it's used for protecting during radiotherapy. A lot is used as injectables for, the article says facial corrections, but a lot of it's plastic surgery, removing wrinkles, things like this. Hyaluronic acid is used largely, and collagens. Spinal fusions, delivery of things like BMPs and OP1 are delivered by a lot of natural polymers. And then advanced heart failure, there's a system of alginate used to try to help the heart muscle remodel. So just to give you an appreciation for some of the applications. All right, so now let's talk about some of the ways that we've thought about, and I'll kind of walk you through some of the history, a little bit of my lab, picking different vignettes that might be of interest. So one of the things we ask about is when you're picking this hydrogel and trying to design a product, how simple is complex enough? You don't want to try to make the most complex system to regrow a tissue, but how can I make a simplified product that still works well? So when we began in this, it was a time when the autologous chondrocyte implantation therapy was just starting. And so we thought, well, one of the reasons autologous chondrocyte implantation maybe doesn't work so well is because they were just injecting the cells back into patients' knees. We thought, well, this would be a great way to put a hydrogel into place to recode or fill a defect in the knee along with the chondrocytes. There's an obvious source of cells. And so if we have the cells, there's nothing defective about the cells. They can synthesize and make tissue. It's just that cartilage is avascular and not innervated, so it has limited capacity to regenerate. So we were designing different types of PEG gels, and we wanted to be able to encapsulate or co-deliver chondrocytes with this. And if you're interested in the chemistry, that's PEG. And so we put different types of reactive groups on PEG that when you expose it to light, they covalently link up. So you suspend these PEGs in with chondrocytes. When you expose it to light, it solidifies and forms a gel. So what we wanted to be able to do was, so at the time, there's microfracture surgery to try to get cartilage to regenerate, drilling into the subchondral bone. A lot of times that leads to fibrocartilage. Can reduce patients' pain, but the cartilage is going to wear away. Autologous chondrocyte therapy was going on at the time, but they were just growing up the cells in a bioreactor, injecting it, and then taking a piece of tissue from the tibia, the periosteum, suturing it in place, injecting the cells underneath it. And so it was really debated, does this work very well or not? Some people thought it did, but it worked best for small focal defects and not large defects, so it had a very limited patient population that was serving. And in fact, autologous chondrocyte implantation still exists today, but Genzyme, which is now bought out by Sanofi, is finally making money off of this. So they invested all of this to get FDA approval for this cell-based therapy, and now it's just starting to take off some. And it uses a hydrogel carrier for it as well. And so the idea is to have an alternative to joint arthroplasty. All right, so this is probably too much, but I'm an engineer, right? So we like to do calculations. And so we were kind of armed with this idea, like if you just look down here, what you can control about a hydrogel is all the linkers between it. So it's like a three-dimensional mesh in space. And that controls everything about the properties of the gel. So if I want to put this in somebody's knee joint and I want it to be able to withstand certain loads, I have to be able to get these linkers right. But if I cross-link it too much, then the cells aren't going to be able to survive in this, or they're not going to be able to secrete and deposit the collagen and the other components of the cartilage I want to regenerate. So we did all these calculations and optimizations and a whole bunch of experiments, and I'm not going to go through all that. Instead, I think I'll just get to the punchline here. This was a couple of students' theses and optimization of figuring out how we could design these hydrogels so they had the right properties and they would break down at the right time. Our linkers would break down. And here is an example. So these are histological sections. So these are all the chondrocytes. They're in these lacunae. They have the spherical morphology in cartilage tissue. And this is the collagen that they're depositing. So we're staining for collagen. This is one where the hydrogel is breaking down way too slow. So the tissue isn't forming uniformly. This is one where our hydrogel degraded way too quickly. And so it just left big voids and it fell apart too quickly. And then we figured out all different kinds of ways to optimize this so we could get uniform tissue. The right type of collagen is also really important. It's type 2 for articular cartilage. It's one of the few tissues with type 2. But after all that time, and then we collaborated with orthopedic surgeons, and we went all the way up into a sheep model, or a goat model, excuse me, and we made defects in their articular cartilage, and we could design formulations where we could regenerate the cartilage. So that was pretty cool. So this is what the native goat's cartilage looked like, and this was our regenerated cartilage. So that took about 10 years, going through all that bench, through all the animal trials, and it did go into humans. So there was a European clinical trial, and they followed patients for a year, and following their MRI fills and some of their pain scores and mobility scores. And so this was really exciting. It was first time in humans, but it was also maybe a lesson learned some too. So we didn't think about or know as much about reimbursement for procedures. This also happened, this clinical trial was going on in 2009, and that was right when the stock market was crashing. There wasn't as much venture capital invested in this, and this is a really expensive trial to do. So, the technology works. We can regenerate cartilage. This ended up being bought out by a company, Biomet, that hasn't gone through the next stages of clinical trials. But other companies had, and I think for us it was somewhat of a lesson learned that at the time, like the FDA, how do you get these approved? They're all combination products. You have a biologic, a cell. You have a device, a material. And we learned a lot through this process. But it also then brought us back to thinking about different types of cells we might work with, different ways we could make impact, because we learned a lot in this first study. So the one thing I want to tell you about those PEG materials that, how simple is complex enough, is that those materials didn't give any signals to the cells, right? All they did was provided a three-dimensional physical registry so that the cells would stay in place. They might get the right mechanosensing if someone were to ambulate or walk. But the cells were doing all the work. They were synthesizing everything, and the materials just getting out of the way and degrading. So as we began to think, so at the time we were thinking things like, well, what if we could do something for microfracture? When you do microfracture, the bone marrow comes up into the space. What if we could make a hydrogel that those bone marrow cells could infiltrate into, and that instead of getting fibrocartilage, it could be articular cartilage? And that would be simpler, right? Because I wouldn't have to have a cell delivery system, right? I could just design a material to signal to those endogenous cells. So that's kind of what we were thinking about at the time. But the thing about our PEG gels was that, so these are the chondrocytes in the PEG gels. Cells don't recognize PEG chemistry at all. So it's really a good system for cells that maybe are already terminally differentiated, that they don't need to interact with their matrix a lot. They're also really good for promoting cell-cell interactions. It's like if you wanted an islet delivery system, or you had cells that you wanted to grow into clusters so they interact with each other because they can't interact with the PEG very much. So, but here's what happens. We started working with these bone marrow cells. So here are these bone marrow cells, and we started putting them inside of our gels and put them under a microscope. So here's just two cells, this is a low resolution image, but so these cells, they're anchorage dependent. They get a lot of signals from their bone marrow niche. They're trying to attach to our PEG, and they don't see anything. So they are secreting tons of adhesive proteins. They're trying to move towards one another, but our linkers aren't degrading fast enough on their own, and they cannot degrade those linkers. So these cells, in about 48 hours, would undergo anoecus, a programmed cell death because of lack of matrix interactions. So we had to begin to think about different types of ways to change our hydrogel. So here's what we had. That's our PEG, put chondrocytes in them, pre-engineer it so it would degrade at certain rates just by hydrolysis. I could pick if I wanted it 2 weeks, 4 weeks, 6 weeks. We were pretty good at that, but we didn't know enough about these bone marrow cells. So we began to think about, well, how can we make our hydrogel be more like a synthetic mimic of the extracellular matrix that cells reside in? So we began to think about, do we need to put in hanging groups, functional groups that cells could bind to and get a survival cue? Could we put in linkers that the cells could degrade? And The ECM, too, is also a reservoir. It stores lots of growth factors. So could we even think about ways that we could have little affinity binders that could store different types of growth factors to signal to the cell? All right, so that's the way we began to start thinking about this. And so in our way, we're good at making molecules. And so for us, we thought of how do we mimic this extracellular matrix when we're not really good at assembling and making large proteins and getting them to assemble and form. One way strategy would be you could assemble a perfect collagen matrix, have it be embedded with the proteoglycans and aggrecan found in the tissue. If I could assemble this all together, I just have cartilage, right? I could start with cells and make all this. So what we began to think about was well, how could I mimic some of the key features of the ECM? My hydrogel already has the high water content, and it also has some of the mechanical signals that cells get from their surrounding environment. So we wanted to put in linkers that cell-secreted proteases, like MMPs and other things, could degrade. So we were thinking about linkers like that. We wanted to put in adhesive proteins or have something like an adhesive protein where cells, their integrins could bind to, survival cues. And then we also wanted to think about, well, how could we store different types of growth factors of interest? And so I'm at the BioFrontiers Institute, and Marv Carruthers is a faculty member in chemistry and biochemistry, and he started Applied Biosystems. Applied Biosystems is ways that you can systematically make peptides. They make these instruments that you can just program in which amino acid sequences you want, and it'll automatically make this for you. He designed all the chemistry for that. So at the time, from structural biology, we know a lot. So for adhesive proteins, we know exactly the regions that many integrins interact with. Fibronectin, we know exactly where it interacts with laminin. We know exactly where it interacts with collagen. So we could make these short peptide sequences that would bind to integrins. So RGD is one of them that's found in fibronectin. We also know regions in collagen and other molecules that get cleaved by certain matrix metalloproteinases. So we could make these short sequences and then put those into our hydrogel. And then there are also all kinds of ways that you could use phage display or different types of screens that I could make libraries of peptides and see which ones would bind my growth factors of interest. So this one happens to bind TGF-beta. So some of our research involves making hydrogels that sequester different types of growth factors. So then the key is, so what chemistry am I going to use? And this is one that Balaji Srihar also worked on quite a bit in our lab as well. And so what we did, the chemistry is called the thiolene reaction, and it's also photochemical, which is one of the things we like. So just maybe have your attention down here. This is cysteine. Cysteine is one of the amino acids with a thiol. So we thought if we could make any peptide sequence that we want, if we just incorporated cysteine on one or both sides, I could use thiol as one reaction partner. So then the key part was for my PEG molecule, I needed to find an end group that would react very well with this cysteine. So it turns out that this ring-strained ene was one that we found that when you mix these two together, they don't react at all. But when you expose it to light, you can initiate a reaction that clicks these two together. And so this in the chemistry world is something that's called a click reaction because they proceed highly efficiently. If you have a complex milieu of proteins and cell culture media, or you're putting this in the body, these two reaction partners will find each other and click together. So that's what we used. And so then the idea is this. We used our same PEG schematic where I could control how many arms are on my PEG, how high is its molecular weight, and that was going to let me control the physical properties of my hydrogel. And then now what I could do is make whatever peptide sequences I wanted and introduce that into the mixture, and it would click in at whatever concentrations I introduced. So we We got better with some of our equipment and abilities. So we use liquid robotic handling systems. And you can change the composition across multiple samples, and then you just expose it to light with your cells, and it all gels. And then you can put it onto a high throughput. We use an Operetta imaging system that will automatically take images. And then you can look for hits of what's interesting. What property do you want? Do you want the cells to proliferate really fast? Do you want to increase their secretory properties, so secrete tissue really fast? And so here's just one system that these are these same bone marrow cells we were interested in. And we took a forearm peg and we clicked in an adhesive sequence and we clicked in a degradable sequence. And now this was high throughput, so this is a low magnification image on the top. So all these small dots here, those are the bone marrow derived mesenchymal stem cells. And now it's dramatically different, right? They're attaching, spreading, migrating through the hydrogel. And if I just misspell this tryptophan and instead insert an alanine, the cells can still attach through the RGD, but they can't degrade it very well. So they're stuck inside this gel because it has less susceptibility for cleavage. So now you can even begin to start thinking about putting hydrogels into wound environments so certain cells could infiltrate, but not others. So keep macrophages out or allow just bone marrow cells, such as the MSCs, infiltrate. Okay, so one of our first tests for this was I had a fellow come into my lab from the med school from otolaryngology, and he was really interested in designing a material system to help promote fast bone regeneration when you have large craniofacial defects. So you want to regenerate bone fast, so if you had a car accident or if you had some congenital abnormalities and you have to have multiple surgeries to fix this. At the time, they were just using bone cement, which wasn't so great. So he was testing out, so he made these critical size defects in a rat skull, 9 millimeters, so it's large enough that it won't heal on its own. And then he put our hydrogel systems in here, and first thing, we just wanted to see what was going to happen. Would it degrade? Would it impede healing? And then eventually we wanted to deliver like BMPs or something to the site. But it turned out that putting in our hydrogel, so this one I call permissive, that's that alanine variant. There was a little bit of mineralized tissue regeneration. I'm not going to show you the analysis, but it's not quite mature bone, but we get some bone regeneration here. But here, when we just put in our MMP degradable material into this wound environment, we were shocked that we saw lots of regeneration. So we think what happens is that our hydrogel helps stabilize the clot and all the factors that are present because of the wound and the bone marrow in that area. And then when it stabilizes and the resident cells can infiltrate it, that led to some of the regeneration. Now, it wasn't perfect. It still took 8 weeks. But this is just a material influencing regeneration. But that kind of left us with the point of, so if you wanted to improve this, there are all kinds of questions you could answer. Again, this question, can I introduce complexity or do I need to? And so you might be asking, well, maybe I should deliver a chemotactic factor to cause cells to infiltrate faster, and then maybe I want, when they do arrive, that they get an osteoinductive factor, like a BMP. Maybe, what if I delivered MSCs along with this? Would it regenerate faster? Could I better signal to endogenous cells, or do I need to personalize this? You know, is it going to be different depending upon the size of the wound? Or is it going to be different if it's a small child versus a very elderly patient? So those are just some of the questions that we ask. And so one of the ways that we've sort of taken this is we don't just make these bulk monolithic hydrogels. So this is the way we're kind of thinking about this. This is current research going on. Instead, we make these little tiny microgels. You could even inject through a needle if you'd like. And that what we envision is that these microgels when they come into contact with each other, if I mix two different populations, they have those complementary reaction pairs, and they'll click together and covalently bond. Now, what's interesting about this to us is that I can make, off the shelf, have a whole bunch of different particles, microgels, that I could pick and say, I'd like to really include some adhesive ligand, or I'd like to deliver a certain type of growth factor and a certain type of cytokine or a certain type of chemokine. I want it to degrade, some of them fast, some of them slow. It's kind of these off-the-shelf ways you can begin to think personalizing biomaterials. And so this is just sort of what they look like. So we false-colored labeled two particles so you can see when you mix them together, you can control the size, so you can control, if they're small, you get less interstitial space, less porosity. If they're large, they're very open. And then you can co-assemble these in the presence of cells, too. So you just inject them or spin them down. And so you can also begin to control, are cells connected to one another? Are they interacting just with the matrix? Or are there lots of cell-cell interactions? And I'll tell you why I think that's interesting or important. So since we've started working with these bone marrow-derived MSCs, they are probably the most common stem cell used in clinical trials. It's quite amazing all the different ways that they're used. So bone and cartilage at the time, the thinking was very obvious that they're local, they're found in that tissue. They can undergo differentiation to become osteoblasts, and they can undergo differentiation to become chondrocytes. But people were starting to inject them into hearts after heart attacks, into kidneys, trying to control different types of fibrosis. So there's a really interesting thinking in the field right now that these cells, that perhaps one of their big benefits are all the factors they secrete. So it's more about their secretory properties, whereas a while ago we were thinking all about their differentiation potential. And so here's the explosion in the clinical trials. And I just looked up, there's more than 500 now throughout the world. I think 300 of them are in the US, these clinical trials. But what's interesting about this is that, so when you do a bone marrow aspirate, these cells are rare. They're only a small fraction of the cells from a bone marrow aspirate. So because of that, you have to expand them. And the ways that they're expanded is they have little beads made of silica that they attach to grow them on, and then they put them in these personalized little bags, and they circulate fluid through them, and they just have to grow up to clinically useful numbers, usually 1 million to 10 million cells per treatment, if not more or something. So the thing about that, and the reason that's interesting to us, is that how you expand these cells totally changes their secretory properties. So we've shown expanding these cells on our little microgels. They'll have totally different secretories than if I culture them on a stiff silica bead. So that's one thing that's interesting. Another thing that's interesting from us is that in most of these trials, they inject the cells, but within 24 to 48 hours, most of them are either just washed away or they die and they don't stay local. So there's also a need to promote their survival and sequester locally, so that also made us think about ways we could design material systems for this. All right, so I think I already mentioned this. So let me tell you just, this is not here for the details, but told you that their secretory properties totally change if you put them on a soft gel versus a stiff gel. Another thing that totally changes their secretory property is how many nearest neighbors of MSCs they have next to each other. If they're single cells, they have dramatically lower secretion of factors. But if I can cluster them together in groups of 5 to 10, dramatically elevates their secretory properties. And you don't want to get too big because then the cells in the middle die. So this is all this is showing is that we did studies where we made our microgels of 200 microns, 100 microns, and 10 microns, and then we just did cytokine arrays of all the factors that they're secreting, looking at all the growth factors and what was different about this. And it was quite dramatically different. So I think that's something that's important and for the future. And then this is one of the ways that we're using these more complex systems is we're looking at things like, this is an even harder defect to fill where I have a large critical size defect in a femur. And this is just how it all looks. We can deliver MSCs to these, you can inject them, and when they do, they just form these three-dimensional structures that click together. So you can shape it exactly to this. And we're beginning to learn better ways and which factors to include, right? So it's kind of a nice bench to in vivo, back to bench. All right, I think I'm just about out of time. So maybe I'll just make a small point. So in my world, we spend a lot of time, a lot of people don't believe in the in vitro models, right? So if it doesn't happen in a patient, if it doesn't happen in an animal, I'm not getting very useful information from in vitro models. So we spend a lot of time trying to test that and see if our models are any good, or if we're just artificially measuring things that happen in vitro in a hydrogel. And I think we have some good examples that this is. But there's also a lot of interest in this idea or concept of precision medicine and what becomes more precise medicine if I'm using a patient's own cells, designing material systems for that. But is that really helpful or not? Is that hyper hope with what's going on? And if so, could biomaterials play a role? So, for example, in the cancer field, there's lots of interest in designing biomaterials, little nanomaterials that can target the cancer cells, find it, deliver the payload in the right way. And about a year ago, there were a whole bunch of reviews showing that no matter what you did with these, you didn't improve things very much, that it didn't target and things. So that led to a lot of this hype or hope concept. And so we try to think about ways and try to pick examples where device customization might matter. One of my collaborators that we have a cardiovascular project on is, her favorite term is a woman is not a small man. So when we start thinking about giving drugs and doses that are just based on weight, it can be very, very different. So women develop heart valve disease, but theirs tends to be very fibrotic, and men develop heart valve disease, and theirs tends to be more calcific. And right now, the only thing we do is we replace the valve. We don't have ways to treat it, but obviously men and women might be treated very differently. So with that in mind, I might skip some of this, but I heard that several of you might also work in neuro and work with Parkinson's disease. And so one of the things that's very preliminary, and we've only done a tiny bit in this space, is how could you design hydrogels as personalized in vitro cultures to better understand disease progression. And so I'll skip through some of this motivation. But so we've done some limited work with dopaminergic neurons. And many of these come from either ES cells or from fetal tissue when they're transplanted. And even though they're differentiated to dopaminergic neurons, they're pretty immature, and you're putting them into a brain environment that's usually an adult brain. So we began to think about, are there different ways that we could incorporate different types of factors into the hydrogel to promote survival and promote longer-term function? And so this is, I guess, one tiny other example where we do some of making little nano-sized particles that we can load up with different factors inside of here, and this is sort of what they look like, and we put in growth factors and different cytokines and chemokines. But we've done a lot where we've incorporated light-triggerable linkers into our hydrogel. So rather than it being degrading by an enzyme or degrading by hydrolysis, we put in these light-degradable linkers. And so now when I put this under the microscope, right, so I have my dopaminergic neurons, and then I have all these depots around it. And then I can, with my laser and a confocal microscope, come and address those depots. And when I hit it with light, they'll very rapidly degrade and release their contents. So we're really interested in using this as a model. If I have a culture of, for example, a neurosphere culture model, and I begin to release different factors, can I look at survival? Can I look at axonal outgrowth? And so I had one postdoc who had a lot of experience with fetal rat midbrain precursor cells and culturing them as neurospheres. And so we did all kinds of things where we put factors around here, and we looked at how we could control axon outgrowth. So this we're just sort of trying to understand, can we go from something like this to actually controlling and directing where an axon would go? Okay, so thanks for, again, the invitation. I hope some of what I told you was of interest. I think what I tried to maybe show is that there are lots of different types of biomaterial systems that are very compatible with delivering cells in vivo, culturing them in vitro. We're getting better at designing bioresponsive materials and ones that can be targeted to promote tissue regeneration or allow for making advanced tissue models for drug screening in other areas. But I do think there's lots of opportunities to bridge current and more advanced biomaterial systems with clinical needs. So all this work, I guess, was done, none of it by me. So since I kind of went through time, the current lab members that work with the MSCs here and bend the cartilage, and a couple of postdocs who do a lot of the chemistry, and then Balaji Sridhar was a former PhD student, and we still collaborate some. And these are former group members. Pete did a lot of the bone regeneration model. Melissa did the work with the neurons, and Ben, Mark, and April are also chemists that developed a lot of that thiolene reaction that we use so much. And so, thank you, and I'd be happy to answer questions. Yeah, I think that's the tricky part, is how much force you can apply. So I think some of those physically associating polymers work pretty well. Before they react, they have, it's almost just like water. You wouldn't be able to tell the difference of how it flows or what its viscosity is. It's only when they're starting to associate. So I think we're beginning to also think about ways that you could either have something in situ react after you've injected it. Another way we're thinking about is, can we make these microgels really small? So I don't know how big of a gauge needle is practical for some of the applications, but that's the crux.
Yeah. With what you were saying about the stem cells' main function maybe being secretory, is there any value in using the little plug-and-play hydrogels just to deliver those same growth factors rather than deliver the cells that then secrete the growth factors?
Yeah, I think that's a great point, and one way we're thinking about. So we're at the stages where we're analyzing a lot of the secretome and doing a lot of proteomics to try to figure out what it is. So you can see what's going up and down, but sometimes you don't know if it's a small molecule that's in low concentration that's really potent compared to another. So it's figuring out some of those combinations. But I think that's what will work long term, because if you can get rid of the cells and just deliver those factors in the right concentrations, it's just that we're probably a little farther away from knowing what those combinations and factors will be. We have some ideas. But
hyaluronic acid and stuff for like knee osteoarthritis.
You all probably have a better opinion than me. So I think mechanistically, I don't think we know enough about how it works. So I think people try to argue when it breaks down in different molecular weight fragments can influence activities of cells, but it's just because I don't understand how it works doesn't mean it doesn't. But yeah.
You mentioned the clinical trial being on hold for the chondrocytes. If you sort of had to predict what will come to sort of the clinics first, you talked about heart, you talked about brain, you talked about all sorts of injections. Where do you think it's going to hit first?
Well, so far it's been in areas where it's restoring mostly mechanical integrity. So the very first, some of the very first cell-based products that we made were engineered skin, and mostly because that could save lives, and so they were cell-based products of tissue engineered skin that was used. And then I think the next advancements came in cartilage, and so there are products that exist. It's just that more of them are the patient, they aren't insurance reimbursed, so it's more expensive, and it just kind of depends on your lifestyle. The bigger blockbusters haven't necessarily used cells, but all the recombinant BMP that was used, although that also had a really bad time. Medtronic it was used off-label for spinal fusions, and when it was done in the cervical region, it caused huge inflammation and cut off airways. So they had to do a quick recall, and I think that's an issue of dosing isn't right and how you deliver it, because the amount of BMP they deliver in their infused product is probably, if you had 200 people in this room and you took all the BMP out of their bone marrow, that would be the one dose they give. And it's just because it's not very efficient, it just doesn't stay local, it goes all into. So those have been some of the biggest. There has been some work in trying to regenerate heart valves, especially for young children, because young children, they're growing so much, so they have to have valve replacements many, many times. And some of the tissue-based valves that are used in younger patients, they calcify. And so then that's largely, a mechanical area. Those have been some of the advancements. And then I guess the last one is that they've also worked with tissue engineering blood vessels. And what's kind of interesting for that is, so that's actually being used for patients that are on dialysis, and they're using that as part of the port because their veins, I guess, collapse from lots of. Then I think, it's hard to say, but you almost have to pick something that's a very debilitating disease where the treatments are so limited. And that's, where I think there have been some advancements, like the Edmonton protocol got approved because patients' diabetes was just so out of control. They had already had I think maybe a kidney transplant, so they were on immunosuppressants, and so that's where that started. So I think perhaps, in some of the space, as we begin to understand more about neurodegenerative diseases, there might be some. There's also some really tough areas too, like with spinal cord injuries. It's like the tiniest nick in your spinal cord, and we don't know how to repair and regenerate that. So there may be some ways coming with that, because that would have a tremendous impact on life. But even that, I think that was one of the very first clinical trials for human ES cells. Was they had differentiated them to, I think it was oligodendrocytes, and they were injecting them to certain patient populations, and they stopped the trial. So that was kind of a long answer and not maybe quite so direct, but thank you.